We will continue in the second session to pick up themes that were already raised in the first session. Just to retread a little bit of history, we knew that cancer progression was a multi-step process already starting from early in the 20th century because of histopathological studies. By 1999, there was a correlation between genetic evolution and histopathological evolution, and there arose in some people's minds the notion that multi-step tumor progression was an evolutionary process very much akin to Darwinian evolution, albeit occurring in the microcosm of a tissue. But then, over the last 15 years, things have gotten to be vastly more complicated, because we've come to realize, if you listened carefully to this morning's session, the talks we've already heard, that often the rate of mutation greatly exceeds the rate of Darwinian selection, so we end up with multiple coexisting clones, and that's only beginning to reveal the extent of intratumoral heterogeneity, as we'll hear from analyses of individual cells, which actually differ from one another. Moreover, the talks we heard this morning were focused very largely on the genomes of cancer cells within a tumor, and there's an entirely additional and possibly much more complex dimension of variability within tumors, and that is changes in epigenetic programs, changes in transcriptional programs that may flip cells quickly from one state to another and may profoundly affect their biological behavior. Our first speaker in this vein is Kornelia Polyak, who got her MD in Szeged, Hungary, PhD at Weill Cornell, working with Jean Massaguet at the Sloan Kettering, and then did a postdoc at Johns Hopkins working with Bert Vogelstein, and she is now a full professor at the Dana-Farber Cancer Institute and Harvard Medical School, and she's going to tell us about breast cancer evolution.
Right, so first I'd like to thank the organizers for giving me the opportunity to present some of our recent work in trying to understand tumor evolution in breast cancer. And the first part of my talk, I show you that we have a really translational lab. We go from clinic to lab and backwards, and we're developing technologies to study intratumor heterogeneity in breast cancer. And then the second part, I show you we're also trying to understand the functional relevance of this intratumor heterogeneity. So I don't have to give a long introduction, but this one is for Bob, maybe to update her book, because diversity or heterogeneity is really a hallmark of cancer. And there are many levels of heterogeneity in cancer that differs from normal cells. There have been a long discussion this morning already on a genetic heterogeneity. Normal cells are homogeneous and tend to be genetically normal. Tumors are genetically heterogeneous and highly variable. The microenvironment in a normal tissue is very organized and structured. Every cell has a place in a body. In contrast, tumors have a very distorted microenvironment. In normal cells, the cells have a very defined cellular state, and the barriers between these states are very high. The cells don't switch cell fates very easily. In contrast, in cancer, there is a very high degree of phenotypic and cellular state instability. And as a consequence, normal cells have a very robust architecture and network architecture, and tumors are very messy. Basically, almost anything we can measure in a tumor, we are likely to find heterogeneity. These include the response to treatment, epigenetics, genetics, and then various abilities, functional properties of the cancer cells, which are somewhat harder to measure, and that's why it has been somewhat behind with the genetic alterations. So just to kind of contrast this kind of classic textbook point of view of how we have liked to see tumors progress, there's always like the suggestion there is a clonal succession and the dominant clone is acquiring additional changes, which doesn't seem to correlate with the data we see in more and more tumors, where we see a very extensive subclonal heterogeneity and actually see subclones disappear or reappear during the tumor evolution. So we have been studying this intratumor heterogeneity for a number of years, and one of the measures that we have borrowed from ecology is actually to be able to have a quantitative measure of tumor heterogeneity is this Shannon index of diversity, which I just briefly explained. It's basically ecologists use it to calculate biodiversity in a particular ecosystem. And it's a formula that's fairly easy to calculate. You just count the number of different species you see and the individuals in the different species. And translating this index to cancer cells, what we have been using is basically a feature that you can measure in a cancer cell, a unique feature, and then the commonality of that feature is what a species is. So for example, a cancer cell with a particular mutation, you can call it a species, and then you count individual cancer cells with that property, and then you can define this Shannon index. And in the past, last year, we published a paper that I just briefly mentioned. We have been using in situ technologies that allows you to look at genotype and phenotype at the same single cell level and in intact tissue samples. This particular study, we used immunofish, where we combine immunofluorescence for markers such as CD2444, Ki67 for fish with copy number alterations. And just to summarize of this particular study where we looked at pre and post-treatment neoadjuvant chemotherapy samples in breast tumors, correlating with many other findings, we basically found that tumors that had the lowest pre-treatment diversity seem to have the best response to treatment. In the case of neoadjuvant chemotherapy, this is a pathologic complete response, so only the patient that had the lowest diversity reached pathologic complete response. And this is a brief summary of this paper, but again, due to time constraints, I don't have time to go through all the details that we have concluded. So what I'd like to show you now is a new technique that we have been developed and applying to tumors, because in addition to copy number variation, we also would like to look at the point mutations in the tumors and see which point mutations occur in the same cell versus different cells, and what is the topologic distribution of these cells. So this technique that was developed by Michalina, a very talented postdoc in the lab, has, we call it Starfish. Starfish stands for specific to allele PCR fish. It's a combination of in situ PCR and fish for copy number alterations. And we have been collaborating for a number of years with Francisco Micor's lab, who's a mathematician at Dana-Farber, and this Lin Liu, who is a student in her lab who collaborated on this study. So what is Starfish is basically, we're doing an in situ PCR, so we do a PCR for a mutant and wild type allele or a particular gene, and then do a detection with fluorescent probes, and at that point we also combine it with copy number probes such as HER2. And that way we can get single cell data for a mutation and also copy number alterations. We developed this technique for PI3 kinase mutation, the various PI3 kinase mutations. The H1047R is the most common mutation, and in HER2 copy number variation, because these two genetic changes are the most common genetic changes in HER2 positive breast cancer. And this is just shows an example of an image. We have the single cells that we image in a confocal microscope, and then for each cell we record the signal for wild-type PCR, mutant PCR, CEP probe, and HER2 probe, and the nuclei, and that we calculate this. When we validated the technique, of course, we want to benchmark it against various other highly sensitive techniques for mutation detection. We compared it to the droplet digital PCR and also the sequenom mass arrays, and we have an excellent correlation to be able to detect mutations in cells where we were assessed, in tumors where we assessed with the different techniques. Then we applied this Starfish to a patient cohort of HER2 positive breast cancer patients that were undergoing neoadjuvant chemotherapy, and we had samples before and after neoadjuvant chemotherapy, and then the patients got adjuvant Herceptin, and then we analyzed the response after the adjuvant treatment, try to associate with changes and response. This particular is a one patient example. I don't know how well it projects, but basically we image the different channels for each color, And then we have a computer program that performs an automatic segmentation of the image and calculates the signal for each cell and preserving the topology. And we can get this classification of the cells into the different genotypes based on the amplification and copy number variation and the mutation. One of the surprising or actually somewhat unexpected finding was that we actually see cancer cells with each 3 different genotype, meaning we have cancer cells that have only the HER2 amplicon, only the PI3 kinase mutation, or both changes. And this is a frequency of the cells for the different genotypes before treatment and after treatment. What you see that in before treatment, the majority of the cells are wild type for PI3 kinase, whereas we see in a post-treatment sample a significant fraction of the cells have a PI3 kinase mutation, showing that these are enriched after neoadjuvant chemotherapy. We calculated the Shannon index that I introduced you, and we calculated this Shannon index 2 different ways. One of them is looking at the changes in species diversity, meaning like if we classify the cancer cells into the 5 different genotypes and then compare pre- and post-treatment samples, and in each tumor we assess this before after treatment. Overall, we saw an increase in species diversity after treatment in a post-treatment samples. But interestingly, the patients who had an increase or no change in diversity, there was no difference in long-term survival after treatment. In contrast, we also looked at the topologic distribution of the species diversity, because in each tumor we had multiple regions of the tumor assessed. And in this case, we also saw an increase in a topologic distribution of genetic diversity. And in this particular case, the patient who had a significant increase of this diversity did worse than the ones who did not have any change. What this could mean that maybe in these tumors, the microenvironments in the different region of the tumor are very different, and they're selecting for cancer cell populations with different properties. Interestingly, we had 2 cases where there was actually a decrease in intratumor, this topologic diversity. Those 2 patients were the only 2 patients who also got neoadjuvant trastuzumab during treatment, and the decrease in heterogeneity could mean or likely mean that there was a pre-existing resistant clone that gets selected by the treatment, and that could explain why this patient had a poor outcome, because they had a pre-existing mutation or resistance to HER2-targeted therapy. Another advantage that we can use for these in situ methods is that you can create these topology maps of the tumors. So this one is the different colors reflect the different genotype of the cells, and the intensity of these the circles, distribution of the circles reflect the frequency of cells with a particular genotype in that region of the tumor. What you see, and if you look at the 3 different areas of the tumor, you see extensive variability, and also the before and after treatment samples. In this particular case, you see significant changes in the distribution of the cells with different genotypes. And because we have the whole topology preserved in a computer, we can actually calculate this dispersion or clustering score in a tumor, basically asking if cells with the different genotypes, are they more or less likely to be close to each other, or what is the distance between them? And in this particular example, what we found that the PI3 kinase mutant cells seem to have a higher clustering score, both the single mutant and a double mutant cells. And this seemed to decrease after treatment, which is in combination with the increased frequency of the cells. It likely means that these mutant cells are more migratory and invasive, and they actually distribute within a tumor. In contrast, the HER2 amplified cells that don't have the PI3 kinase mutation, they seem to have a lower clustering score, and they actually seems to increase, which again likely reflect that these cells are probably not that migratory and invasive, and also the proliferation rate may be different for these tumors. The wild-type cells that are most likely stromal cells had a low clustering score, and this was not affected by treatment. So just to summarize the key findings of the study, and I had to go through it somewhat quickly to be able to show you the second part of my talk, is like what we see that there is a very high degree of cellular genetic heterogeneity in HER2-positive tumors for HER2 amplification and PI3 kinase mutations. We see cells with individual and both genetic changes. We also calculated, based on the frequency of the cells, it looks like that the double mutant cells have fitness advantage, but interestingly, they still don't take over the tumor, suggesting that there is maybe some advantage to maintain this heterogeneity. We also found that the frequency of PRC kinase mutant cells is actually much higher than predicted based on the exome sequencing studies, which thought to be about 40% of the HER2-positive tumors. In our study, 20 out of 22 cases, we can detect some PRC kinase mutant cells, but in the majority of the cases, it's below 5% of all cells within a tumor, which is easy to miss by more bulk sequencing. I also showed you that a PRC kinase mutant cell seems to be enriched by neoadjuvant chemotherapy, which actually suggests that it actually would not be beneficial to apply chemotherapy without HER2-targeted therapy up front, because you're likely to increase the probability of resistance. And then we also found that the significant increase or decrease in the topologic distribution of a genetically distinct cell population seems to predict a worse outcome compared to patients that have no change in this diversity. So the second part of the talk, I also like to show you some data on trying to understand the functional relevance of these intratumor heterogeneity. The reason is because we have heard many talks, and we also see this extensive heterogeneity in tumors, but the question is, why is this heterogeneity is even maintained? If we see that the double mutant cells or the multiple mutations seem to be conferring fitness advantage, why are the minor clones are still maintained in the tumors? And what are the consequences of this subclonal heterogeneity within tumors? Are there interactions among the different subclones? And, or, is it direct interaction or indirect interactions? And then how the treatment may change these changes. And we try to address this by designing an experimental model that basically can mimic subclonal interactions during progression of the tumor. And the approach we used, we took a breast cancer cell line, we screened the whole panel of cell lines for tumors that seem to be consistently forming tumors that not able to progress on their own, but at the same time have high cell turnover in the tumor. So this particular MDA468 cell line seems to form these small tumors that don't seem to grow over a long period of time, like 3 to 4 months that we followed. But at the same time, there is a high proliferation based on KH67 and BRDU assessment, and this is compensated by high cell death. The reason why it's important to have a proliferative tumor, because you need cellular turnover to be able to assess clonality changes in a tumor, and also such a high turnover intensifies the competition among cancer cells for survival. At the same time, because these tumors don't grow and don't progress, anything that can make them grow and progress will be relatively easy to detect. And then we introduced clones into these cells by expressing various genes, factors that have been implicated in cancer. Initially, we started with a long range of factors that included professional oncogenes, and then we narrowed down to about 18 secreted proteins, because we thought that the interaction among clones would be the easiest to explain by, based on secreted protein, studies of secreted proteins. And then each clone we injected in a mice with mixed with 95% of the parental cells. We call these tumors monoclonal, or we mixed all the clones together, and these were the polyclonal tumors. And then we assessed tumor growth, progression, and also when we took out the tumors, we looked at clonality by a PCR assay. First conclusion was that almost nothing makes these tumors grow. Majority of the clones were just as small tumors as the parental cells. Only 2 clones, CCL5 and IL-11, were able to make the tumors grow. These go very rapidly. So these were the only tumor drivers, defining a tumor driver as a clone that can make tumors grow. We also found that the polyclonal tumors have very unique features. First of all, they were the fastest growing ones. They grow very fast. We actually had to terminate the experiment early on because we reached the limit of tumor size. But they were also very metastatic early on, at the time points where none of the monoclonal tumors we detected metastasis. We narrowed down to this behavior to 2 clones that seem to be largely reproducing the phenotype of the polyclonal tumors. IL-11 is a very good driver. You see these tumors are large compared to the parental ones. FIGF is VEGF-D. Those tumors are small, but they seem to be very highly vascularized and metastatic. And the polyclonals are very highly vascularized, large tumors, and very metastatic. And then these these 2 clone tumors seem to be largely reproducing the phenotype of the polyclonal tumors, suggesting that these 2 clones cooperate to drive tumor growth and metastatic progression. We also assessed whether IL-11 is required for the growth of polyclonal tumors, because we knew it's a driver, it's sufficient to drive the growth, but it also looks like that it's required even in polyclonal tumors, because when we had polyclonal tumors, they grew fast. When we left IL-11 out, then those tumors did not grow, suggesting that IL-11 both sufficient and required for the growth of the tumors. When we looked at clonality of the tumors, the monoclonal and polyclonal tumors, it was somewhat surprising that actually the ability of drive tumor growth does not correlate with the clonal expansion, meaning that a clone that is a good clonal competitor is not necessarily a good driver. As you see here, I don't see from here, but there is LOXL3, which is a very good clonal competitor, but those tumors were just as small as the parental clone tumors, showing that LOXL3 is a good clonal competitor. It can take over the tumor, but it's not going to make them grow. In contrast, IL-11 is not a good clonal competitor because it would never become a dominant clone, but in contrast, it's a good driver of tumor growth, suggesting that you can have minor clones that actually responsible for driving the growth of the tumor. If you compare the green bars that are the frequency of clones in the monoclonal tumors with the purple ones that are the frequency of the clones in polyclonal tumors, it's very clear that in a polyclonal tumor, none of the clones are as good competitors as in a monoclonal cases, suggesting that having multiple clones in a tumor limits each other's ability to expand, which we call a clonal interference. Then coming back to IL-11, we also wanted to investigate what is the mechanism by which IL-11 drives tumor growth. IL-11 is a family of it belongs to the family of IL-6 cytokines. It's similar to IL-6, but not identical. It seems to have act on multiple cell types, such as the tumor cells themselves, but also on multiple stromal cells, such as leukocytes and endothelial cells as well. The way we investigated whether IL-11 acts in a cell-autonomous or non-autonomous manner is to try to assess the phenotype of the cells in these tumors. So first we concluded that it looks like the IL-11 seems to drive tumor growth by increasing proliferation. The IL-11 expressing tumors had a slight but significant increase in BRDU incorporation. This just shows the image of this. And interestingly, this was not dependent on the frequency of the IL-11 clone, because when we derived tumors that were 100% IL-11 or like just 5 or 20% IL-11, the tumor growth was still promoted equally, which again shows that IL-11 can drive tumor growth as a minor clone. Then a way to assess whether IL-11 is acting on a cancer cells or in a non-cell autonomous manner is we downregulated IL-11 receptor on the cells that expressed IL-11, and these tumors were still growing just fine, suggesting that it's not acting on the cancer cells themselves. Another way to assess it is we expressed IL-11 in breast cancer cells that had no detectable IL-11 receptor expression. This is the MDA468 cell line, and these other cell lines, even with RT-PCR, we don't detect the receptor. But IL-11 still drives the growth of those tumors, suggesting that it's a cell non-autonomous effect through the microenvironment. We investigated potential mechanisms in changes in the microenvironment that may explain the IL-11 effect. And what we found consistently is that the IL-11 expressing tumors were more angiogenic. You had more CD31 cells. And this was observed at early stages, even at the time when the tumor weight was not significantly different between the parental and the IL-11 clone, suggesting that it's likely to be the mechanism by which IL-11 promotes tumor growth. And lastly, We also wanted to do an experiment asking the question, what would happen if you mix a known driver, meaning IL-11, with a good clonal competitor that is not a good tumor driver, such as LOXL3. In this case, we generated tumors that had each clone alone or mixed together at different ratios, and then assessed the tumor growth. What you see that IL-11 is able to drive the outgrowth of these tumors, and even the mixed tumors seem to be growing just the same way as the IL-11 tumors, which was initially disappointing. But when we collected terminated the experiment and looked at the tumors, what we saw that, the LOXL3 tumors, they were very small, not growing. IL-11 tumors were large vascularized tumors. And the mixed tumors, the different ratios behave differently. The ones that had 1-to-1 ratio, they were still solid mass and fairly large tumors. But interestingly, the ones when we had more LOXL3 injected, they were basically sixth, and the middle of the tumor was totally necrotic. Basically, the tumor lysed, suggesting that the tumor collapsed, potentially due to the fact that IL-11 actually got outcompeted in these tumors by the LOXL3 clone. So LOXL3, because it is a good competitor, outcompeted the driver and leading to the collapse of this tumor and necrosis. So just to summarize this part of the talk, what I showed you, that this clonal or subclonal heterogeneity is maintained within tumors. The clonal frequency doesn't seem to reflect the ability to drive the tumor growth, which kind of questions our classification of the driver just based on frequency, and suggesting that some minor clones can indeed be the drivers. Multiple drivers within a same tumor seem to limit each other's ability to expand, and actually eliminating this clonal interference may accelerate progression. I didn't show you, but when we're applying treatment to these polyclonal tumors, it looks like that the good competitors become even better, meaning like we can favoring the outgrowth of cancer cells with the potentially least desirable features due to treatment. Polyclonal tumors are more likely to metastatize, and it also looks like that there is a cooperation to the metastatic progression among clones, such as a driver and a disseminator clone is not necessarily the same one, and they act together. And we have preliminary data also suggesting that metastatic lesions also seem to be heterogeneous. And interactions, we also done mathematical modeling, which again, I didn't have time to show you today, but it looks like that the cell non-autonomous drivers are what maintain intratumor heterogeneity, meaning like tumors that have more multiple subclones likely to have interactions among clones through the microenvironment or other paracrine interactions, and that's why this heterogeneity is maintained. And lastly, I just like to acknowledge the people who did the work, and we have a very diverse heterogeneous lab reflected in the colors. So the first part of the work that I showed you was largely done by Vanessa Almendro, who was a former postdoc in the lab. The second part by Michalina Janisewska, who is a current postdoc in the lab. And then the xenograph study was done by Andre, postdoc, and Doris, a student in the lab. And we have many, many collaborators. We have a very fun and long-term collaboration with Franziska Mikor's lab on the mathematical modeling of cancer. And I'd like to acknowledge all the other collaborators. We have a new sign that we had to put up on the lab this week, because indeed we have a mixed gender lab, and this comes with dangers. But most of this work that I showed you were actually conducted by female scientists, so they can do work besides crying and falling in love. Thank you.